there was another Dalhousie in Scotland. It's a decently Scottish name, I guess, Dalhousie, maybe. It's in the Highlands. But then I tracked down his poster and was like, Halifax? That's next door. Heck, it's like the only place you can take one flight to St. John's in. We should invite him for a talk. That, and because he has a lot to teach us. Anyway, so Teo's here today. By way of more traditional academic introduction, Teo, which is a citizen of France. So the way the education system works in France, as a young child, he streamed based on his aptitudes to be forced to do a particular type of vocation. I think at 14, he said. They detected an aptitude for math stats or statistics. So streamed him into a university, undergrad, master's kind of program of applied math with the expectation that he would become an engineer, but or some sort of applied math and engineering. But he tells me that that was bringing him no happiness. So he managed to manufacture what they call stagiaires or internships. At the end of your master's, you do 2 of these, which got him out of France and to St. Andrews, and started to apply his statistics background into movement ecology. And from that stagiaire in St. Andrews, his PhD was in Sheffield, which then had some interactions with some movement ecologists that were really passionate about their ideas. And then a postdoc back after the PhD in Sheffield, a postdoc at St. Andrews again. And we're fortunate to have him in Canada as now a applied biostats in the movement ecology kind of idea space, and even right along our Atlantic seaboard. So without further ado, I'll actually leave Teo some time to give us a talk.
Hi, thanks so much for the invitation. Thank you so much for the great introduction. So yeah, I'm a statistician at Dalhousie, and I've been working on the study of kind of animal movement, the analysis of animal movement data for quite a while. And today I'm going to be talking about hidden Markov models, which are a general class of statistical models that are really popular or kind of increasing popularity in ecology. For those of you who were at my small workshop yesterday, this is going to be to some extent more of the same. There's going to be a little bit of overlap, but the main topic that I want to talk about today is some kind of extensions of hidden Markov models that I think are kind of fairly recent, and I think will be really exciting for some ecological applications. And in particular, I'll be talking about a new R package that I've been working on in the last 3 or 4 years called HMMTMB, where we've tried to implement some of those extensions, try to make those models more flexible. And I'll first describe a little bit of kind of the statistical background, and then I'll just show a few case studies using that package to show you the kind of questions that we can try to answer with these models. So a little bit of motivation for those of you not familiar with hidden Markov models. Essentially, they're statistical models that are useful in situations where there is some ecological process that we're interested in, but that we can't observe directly. And that happens quite often. Quite often we have some observed variable that doesn't quite correspond to what we're interested in, but somehow tells us something about what we were interested in. And so maybe the running example in my slides and in, I guess, my career is animal movements, where we have tracking data. So we put a tag on it. Well, I don't do it, but some people put tags on animals and get locations of these animals every 5 minutes or hour or something. And you get a track like this seal here. But what you're actually interested in is what the animal is doing. You're interested in knowing when the seal is foraging, when the seal is resting. And you can't quite, tell from the data directly, but with a little bit of thinking, you realize that actually the movement patterns of the animal depend on what the animal is doing. And so from the tracking data, you can infer what the animal was doing if you know quite a bit about the species. And that's one application of hidden Markov models. Another one that might be less familiar to people in the room is for occupancy modeling, and this isn't really my area, but I think it's also kind of a cool application. For occupancy modeling, what we're interested in is knowing whether a species is present or absent at a certain number of sites. And the data, or kind of a typical kind of data that people collect is, some expert goes out in the field to the site of interest and looks around, tries to find the species. And if they find it, well, you know that the species was present if you assume that there's no, wrong identification. But if the species isn't identified, you don't know if it's because it wasn't present or if it's just that the person didn't see it, right? And so that's the hidden part in that context. The hidden part is that the present-absent status isn't directly observed. So this is a data set that I'll come back to a little bit later, but where you have counts, and the counts are between 0 and 3 because actually they had 3 annual surveys. So each time, if the person detected it, it's like +1. And they carried out this study over 9 years. So for each site, you have 9 observations between 0 and 3. And when the observation is at least 1, you know that the species was present. But when the observation is 0, you don't know if it's that, whether it was absent, and sometimes that's what you might infer. But in other cases, you might, use information from other years, and you might try to estimate detection probability to tell you something about what's the probability that you just missed the species, even though it was there. So 2 kind of ecological applications that are pretty popular. So now I need to spend a little bit of time talking about the statistics, and to some extent, if you can just unplug your brains for a little bit if you want, but I'll come back to plots and applications and case studies later. But the basic idea is that a hidden Markov model includes 2 what we call stochastic processes, and one of them I'm denoting with S, and that's what I'm calling the state process. In the animal movement example, it's the behavior of the animal. That's whatever you're not observing, that's changing over time. And then we have the observation process, and that's how that essentially links the state process to the data that we have. The data that we have might be those locations of the animal. That's the observation process, but the state process is what the animal is doing. So we know that the observation depends on the state, but that's in kind of a probabilistic way. So we're going to have to do statistical inference to actually learn about that underlying unobserved process. And the 2 big assumptions is the first one is that the hidden process is what we call a Markov chain, which is just to say that we assume that the status at time t depends on the state at time t minus 1. So what the animal is doing right now somehow determines what they're going to be doing in 5 minutes or in an hour or something. And the second big assumption is that the distribution of the observation at time t, so the distribution of zt depends on the state at time t. Okay, so that's what's represented by those vertical arrows, and that kind of allows you to learn about the hidden state process from the data that you have. And so we end up with those 2 kind of modal components, the 2 processes. We have the hidden state process, and if we have 2 states, so we could imagine that there's more than 2 states, right? You could imagine that there's as many as you want, but for 2 states, you need 2 transition probabilities, one from state 1 to state 2, one from state 2 to state 1. So those are the parameters of your process. Those are the parameters that describe the dynamics of that, for example, the behavior process or the occupancy process, right? You could imagine that this is present and absent from the site, or you could imagine that it's foraging and exploring or something. So first set of parameters, transition probabilities. And then the second part of the model is state-dependent observation, the state-dependent distribution. So we assume that in each state, the observed data, the observed process, has a different distribution. So the probability of observing a species at a site is somehow informed by whether or not it's present. The probability of an animal moving fast depends on whether or not they're resting, things like that, okay? And so we end up with these, here I'm showing, for example, 2 distributions, one for each state. So that's my observed variable here along the x-axis, and then the distribution of that variable in each state along the y-axis. And one typical way to model this is with some family of distributions that I'm denoting just in general as D here. but where the parameters depend on the state. So there's some parameters that describe the observation process in state 1 and some other parameters that describe the observation process in state 2. Okay, I can't actually remember if I explained what the transition probabilities are, but they're the probabilities of transitioning between the states over one time interval of observation. So we end up with these two sets of parameters. And the kind of questions that I want to talk about more in this talk is how we can infer effects of environmental or other covariates on those parameters. And I'll focus especially on including covariates or covariate effects on the transition probabilities. And this allows us to answer questions like, what is the effect of some environmental condition on the animal's behavior? What is the effect of whatever environmental condition on the animal's presence at a site, things like that. Okay, so essentially it's allowing us to investigate environmental or other drivers of the dynamics of that hidden state process, and it's kind of nice that these models can be used for that, because it's a little bit complicated in principle. This isn't one of those things that you can immediately see from looking at the data. It's not. You need to infer that hidden state process that you don't observe, and then infer kind of how the dynamics are affected by those various drivers. So this idea of including covariates on the transition probabilities of hidden Markov models has a long history. People have been doing this, including in animal movement ecology, for at least 20 years. But we wanted to try to make that more flexible, and so I'm going to quickly kind of describe how we've tried to do that. So here's the kind of flexible model that we came up with, where eta here on the left is some parameter of the model. So we're going to say it's something like a transition probability. Technically, it has to be a transition probability on the length scale, because the transition probability has to be constrained between 0 and 1. But let's say just that, this left-hand term is some parameter of the model. Now we're going to say that transition probability is time varying. We're going to say it doesn't, there's not just one number. It's not just there's an 80% probability of going from exploring to foraging at each time step. We're going to say, well, it depends on temperature, or it depends on resource density, or so now we're going to have a different value for that transition probability at each time step of the data. And the way we represent that time dependence is through the covariate dependence. That I'm breaking down here on the right-hand side into two terms. And if you've seen mixed effect models before, this might look familiar. We have a fixed effect component here in blue and a random effect component in orange. And for each term, we have a model matrix that we're multiplying by some vector of parameters in the same way that we often would for like mixed effect models. And maybe this is nothing new if you're used to these mixed effect models, but the key difference between those two terms here, we have unknown parameters alpha and unknown parameters beta. The key difference is that the betas are treated as random effects, which is to say that we're assuming some random probability distribution for those parameters. And so I'm writing this as a multivariate normal distribution with some covariance matrix Q, and that covariance matrix Q determines the kind of the structure in the random effects, and we can get to pretty sophisticated models using this general formulation. So I'll show two examples of models that can be written in this way and that I think can be useful for ecological applications. The first one is kind of what I might call just like simple random effects or something, or a simple kind of random intercept model. And this is kind of the simplest model that we can write in that form, and I want to show it as a little bit of a illustration. But this is for situations where you want to capture inter-individual variability, inter-individual differences. For example, you have data from maybe tracking data from 30 different individuals, and you want to account for the fact that there's going to be some differences between them. Or maybe the survey data were collected by like 30 different, biologists, and you want to account for differences in like, I don't know, ability or something. And so the way this fits in this general model that I was showing before is like this. So here I have my time varying transition probability on the left-hand side. And I'm saying here that it depends on some fixed effects, which here consists of one intercept, alpha 0, and the linear effect of some covariates that's going to be measured by this alpha 1 parameter. This is just a simple linear term just to show what that could look like. And then the more interesting part is this orange bit, where this is how you would write the model matrix if you wanted to have a individual specific intercept. So what this looks like is just a whole bunch of zeros and ones, and the ones indicate which individual or what group or something is active or is used for each row of your data. So you start from the first individual for the whole first bunch of the data, then you switch to the second individual, and then you would keep going like that. And then each individual has a different beta associated with them, and so that adds a little kind of deviation from the population mean for each individual. And those betas are assumed to follow some normal distribution with a variance that measures that spread around the population mean. Okay, so this is already kind of like a pretty useful model. You can either use this if you're actually interested in the values of those random intercepts, or just to kind of mop up the variation that you know is in there somewhere and should be accounted for. The second example, which I think is more interesting, but is also quite widely applicable, it's penalized splines. And that takes us to the world of like things like generalized additive models and smooth covariate relationships, if you've ever worked on this stuff. But let's say that we want to estimate the effect of some covariate, but we don't want to make an assumption about the shape of the relationship. We can use nonlinear relationships. And the way we model this in a model like this is, here I've kept the same linear term. I've just changed the random effect term. What I've done is that I've filled the model matrix, the design matrix, with basis function evaluations. So what we call basis functions are little simple functions. Often they're just like little bumps that we want to somehow combine to build a more complex nonlinear relationship. And so what happens if we fill this model matrix with basis function evaluations and then multiply by a vector of random effects beta, is that we are basically weighting each basis function by the corresponding beta. And by doing that, your different weights, different betas will give different shapes to that overall black curve. Okay, and you could imagine, depending on exactly how many basis functions you're using and so on, you can have a lot of flexibility in getting very complex relationships in this way. And the reason we use random effects again to model something like this is that we don't want the betas to be estimated separately from each other. And the reason being that we usually think that those relationships are going to be smooth in some way. So we don't think that two basis functions that are adjacent are going to be wildly different. We don't think that one of them is going to be a large positive number and the next one is going to be a large negative number or something, because that would create a very weird kind of wiggly relationship. And so the way we represent this is with this normal distribution, where it doesn't really matter exactly what the S matrix is, but where we have a parameter lambda that controls the smoothness of the relationship. So the bigger the lambda, if lambda is like 1,000, basically the black line has to be a straight line. It has to be a linear relationship. And if lambda is really close to 0, the function can be as wiggly as you want. So this is very flexible, but fortunately we can actually estimate that lambda from data. And so that's kind of how we avoid overfitting and underfitting in like generalized additive models. And we can apply the same idea to the context of modeling parameters of our hidden Markov model with nonlinear covariate effects. All right. So 2 examples that fit within this context. And what really helped me kind of start working on this is that it gets a little bit overwhelming exactly what you can do with a model like this. But fortunately, there's R packages that help a lot with creating the model matrices, creating the covariance matrix of the normal distribution. Kind of all the difficult technical bits have already been done by people who know quite a bit more than I do about this stuff. And so, for example, Simon Wood's MGCV package can be used to do all those things, to specify. So you just have to use a formula a little bit like this, where you tell the package that you want a linear effect of the covariate x. You want a smooth effect of y, and you can specify some basis functions here. And you want a random effect of ID. And by passing a formula like this, which takes very little kind of effort to write as a user, you get as an output all the model matrices, all the kind of the difficult bits. And then within this framework, within kind of the MGCV model specification, you can have all sorts of different model models that I'm not going to have a whole lot of time to talk about, but it's not just random intercepts and like splines and stuff, but also smooth factor interactions. You can have spatially varying effects. You can have all sorts of really interesting models. So this is what I've used, and this is what is used under the hood in that HMM-TMB package that I'll illustrate a little bit later for basically model specification. And then the second part of the second difficult technical bit, once you've specified the model and obtained all those model matrices, is actually fitting a model like this. And if you've worked with mixed effect models, you might know that it's a kind of a difficult problem in general to fit these models. The reason being that the random effect parameters, the betas, aren't treated like regular parameters, so you don't want to just estimate those parameters. You need to somehow integrate them out, kind of account for their distribution, but without actually estimating them directly. And this is done, well, yeah, I'm going to talk a little bit about Bayesian inference later, but so I should say right now I'm talking about the kind of non-Bayesian approach to these models. The typical way to fit these models is to use what we call the marginal likelihood. So the likelihood function is a function that tells you basically how plausible some parameter values are given the data, and you want to find the parameters that make the data look the most plausible, but you don't want to maximize the likelihood over the random effect, so you integrate them out. That's what this operation here is, to get just the likelihood of the fixed effect parameters. But this is actually really hard mathematically. Unfortunately, again, some other people have thought about this and solved this problem in the R package TMB, Template Model Builder. And so what TMB requires is for us to pass that joint likelihood, the function that's inside the integral, that's actually relatively easy to implement, although I'm not going to explain how that's done. And then TMB does the hard work of integrating out the random effects and then returning parameter estimates for the alpha, the fixed effect parameters, and then returning also predictions for the random effects if needed. So the one takeaway here is that essentially this is a difficult mathematical problem, but that it has been, a lot of people have already worked on this, and that using existing software, it's actually not too hard to fit these really flexible models quite fast. And so what we did in HMM-TMB is to essentially combine MGCV for model formulation with TMB for model fitting to allow for hidden Markov models with really flexible covariate dependence. And this was work I did with Richard Glennie, who was in St. Andrews when I was there. He's since left academia, but he was really helpful kind of at the beginning when we get this started. I'm not really going to show a whole lot of code, but I just wanted to make a quick comment about how HMM-TMB is structured. It uses kind of an object-oriented approach where you have different classes for different model components. So there's one class for, by which I mean like an object class in R for the hidden state component, and there's another one for the observation component of your model. And so you can kind of specify those separately in whichever way you want. And then you put them together using something like this into an HMM object that has all the information about all the different components of the model. And then once you have created this HMM object, you can interact with it to do things like fitting the model or model checking or plotting the results and everything like that. All right, so that was the kind of very brief overview of what where we came from and how we approached that problem. And now I'm going to show two or three, depending on time, case studies showing how to, what we can actually do with these kind of models. And the first one, unsurprisingly, is going to be animal movement with these tracks of petrel movement. It's not my data. I found it online on the MoveBank data repository. It's 10 tracks of petrels in the South Atlantic Ocean. And so from 10 different individuals. And we have locations at a half-an-hour time resolution for these birds. And I'm going to say a lot of stuff about animals that I don't know if it's substantiated or not, because I'm not a biologist, but it's going to be more for the sake of argument. But I think they're central place foragers. I think it's pretty clear from these plots that they have some colony somewhere here close to the ice, and then they go out at sea to find fish, or they probably eat fish, and then at some point they come back. So we have these kind of trips, foraging trips over a few weeks for these individuals. Okay, so I'm going to try to model these data using a 3-state hidden Markov model, which is to say I'm going to try to identify 3 different phases of behavioral phases in the tracks. And within each state, I'm going to model the two variables that are commonly used for these data called the step length and turning angles. The step length is the distance between successive points, so it kind of measures how fast the animal is moving. And turning angles are changes in direction, basically, so it tells you how directed the movement is. And I'm using gamma and Cauchy distributions for those. And then to try to understand a little bit better kind of the structure of their foraging trips, I'm going to estimate the effect of the distance to colony on their behavior. I want to know kind of, how far away from the colony they tend to forage and things like that, okay? And I don't want to assume anything about, a linear effect or quadratic effect or something. So I'm going to use a spline, a very flexible spline to estimate the shape of the relationship. And also because I have 10 individuals and I don't want to assume that they all have exactly the same behavior patterns, I'm going to include an individual level random effect for the transition probabilities. So the first output that we get from these models is what we call the most likely state sequence. It tells us essentially for each time point of the data, what's the most likely kind of state that it's a little bit like a kind of a clustering or a classification thing. And so here for each observed location, we get kind of a predicted behavioral state. And so you can see that, the yellow kind of captures the fast directed movement, and then the red and blue is kind of corresponds to kind of slower, less directed movement. We can also look at, these are the state-dependent distributions. So we get 3 gamma distributions for step length, 1 for each state, and 3, I think I used Cauchy distributions for turning angles, 1 for each state. And looking at these is often helpful to think about the interpretation of the states. So here you see that the blue state has very small steps. So probably it's like some slow, maybe a combination of like resting and foraging or something. Then red is like kind of intermediate step lengths, and then yellow is like very, very fast, very directed movement. So here I'm giving kind of tentative behavioral interpretations in terms of like the first one might be resting, the second one searching for food, third one might be transit or something. But this is it's always difficult because these are statistical states, they're not biological states, and so you need to do that work of like thinking about how much they actually match the behaviors that you're expecting to find. Did something happen? And so going back to the question that I had originally about, what's the effect, how is their behavior changing with like distance to colony? Here I'm showing the what we call stationary state probabilities, so kind of the probability of being in the different states at different covariate values over a range of distance to center, right? So the center is just where they start from, and then they travel around, at most kind of 1,000 or 1,500 meters from their colony. And if we want to kind of interpret this plot, well, first of all, I should say that the thick lines are population means, and then the thin lines are the individual level effects or relationships. And so if you just look at the thick lines, it kind of looks like from 0 to 1,000 or something, although there's a little bit of a drop here. They're most likely to be in that fast directed movement state, like in transit, they want to get somewhere and they're getting there really fast. And then around kind of 1,200 kilometers or something, all of a sudden there's a huge drop in the probability of being in that fast transit state, and it becomes much more likely to be in the other 2 states, kind of like looking for food or foraging or something like that. Okay, and then so that's kind of the population level, and then I only included individual level intercepts, which means that as you can see, all the curves have kind of the same shapes. They're only allowed to vary kind of vertically. And this captures, I don't know, I'm not going to attempt to have an interpretation of the inter-individual variability here. You could just think that it's a way to capture the fact that there are differences and it should kind of be accounted for somewhere. So if we'd included a linear effect of distance to center, we might have got something a little bit similar, right? Where you could think that the yellow line would probably have gone down, the other ones would probably have gone up. But it would be really difficult to identify kind of that very sharp threshold that we seem to observe around like 1,200 kilometers or something. One topic that's often overlooked maybe with these models, partly because it's difficult, is model checking. And so I would just want to make a quick comment about simulation-based model checking, which I think is one of the most flexible and kind of most promising ways of doing model checking for these models. Where the idea is you fit a model to your data, and then you simulate many, many, many different tracks or whatever data sets from the fitted model, and you compare the fitted you compare the simulated data to the real data, and any deviations, any kind of patterns that aren't captured by the simulated data are basically features that the model is missing, and that might not be a problem. Any model is going to be, lacking something, and so you might be happy with that if that's not the focus of your analysis. But it's always interesting, at least, to check that the particular features that you wanted to capture in the model are captured well. So here, I've done this for 4 metrics: the mean and 95th percentile for step length, and the mean and the 95th percentile for the distance to the colony or distance to center. And there's a function to do this in H2MTMB where you just tell basically the function what metrics you want to compare between the real and simulated data, and then it runs simulations. And what this plot is showing is histograms of the simulated values. So you get a histogram. I think I ran it maybe for 200 simulations, so 200 values of whatever metric I'm looking at. And the vertical line is the observed value. And essentially what you want is you want the observed value, the real data value, to be somewhere in the middle of the distribution of simulations. Because if it's far in the tail, you can even think about it a little bit like a way to get a p-value or something. If it's far in the tail, it tells you that that is not captured well by the model. So here it looks like, at least according to these 2 summary metrics, mean and 95th percentile, the step length distribution seems to be kind of captured pretty well by this model. But if you look at the bottom left plot here, the mean distance to center, in the simulated tracks, it's always between 400 and 800 or 900 kilometers. Whereas for the observed, it's somewhere more like, I don't know, maybe 1100 kilometers or something. So it means that essentially the simulated data don't spend enough time far away from the colony compared to what the petrels actually do. And I think that kind of makes sense, because the petrels, if you remember what the tracks look like, well, you don't have to remember. They really dash really far away, and then they spend most of their time kind of far from the colony. But there isn't really any mechanism, any component in the model to capture that very explicitly, kind of the structure of their kind of the detailed structure of their foraging trips. And so I think what the simulation does is that it ends up, it doesn't go far enough, and then it starts foraging a little bit too close, and then it doesn't end up, traveling as far from the colony as the real data. And in this case, I don't actually think it's particularly damning for this model. I'd be pretty happy to use this model still for quite a few other things, because I don't know that was the focus of the analysis. But in other cases, you might find that there's a problem, and try to think about how to address it by changing your model formulation. All right, so that was the movement example. So let's see if I have time for this one. Yeah, probably. So let's turn back to the occupancy example from before. So it's the data I showed on my first slide. It's from Crossbill, I think. Yeah, Crossbill in Switzerland. I think, again, it's not my data. I'm very grateful to everybody who shares their data freely, because I rely on that a lot. And what we have is, again, 9 successive years. And on 9 successive years, people went to 267 sites. So I'm just showing 6 of them, but there were actually hundreds of sites, 3 times a year. And they counted, how many detections there were. So it's a number between 0 and 3. And I'm going to model this with a 2-state hidden Markov model, where the 2 states are either absent, the species is absent from the site, or the species is present at the site. And what's kind of nice with this model is that the transition probabilities really have, I think, a biological interpretation. That if you go from absent to present, it's like colonization probability. The species wasn't there, and now it is there. The other way around, we call it the local extinction probability. The species was present at the site, and now it's extinct from that site. And within each state, I'm going to model the data, the count, with binomial distribution. And I won't actually talk about it a whole lot, but one nice thing about this is there's a probability parameter in the binomial distribution. And in this context, it represents detection probability. And I'm only going to look at covariate effects on transition probabilities, but you could think about also including covariate effects on the detection probability. Like if it's foggy, there's a lower detection probability, and so on. And the environmental covariate that I have here is elevation, and I was interested in looking at the effect of elevation on those transition probabilities. Okay. Again, I don't want to make any assumption about the shape of the relationship, so I'm just going to use a nonlinear effect. So in this case, when we look at the kind of the state classification, what we get is essentially the model's best guess at whether the species was present or absent from the site on each year. And when we do have an observation, because here we assume that they don't make mistakes, when we do have an observation, we know for a fact that it must be in the present state. But in the bottom right, again, I have another example here where the model predicts that the species was present, but just was not detected. And by the way, I think the reason that they have multiple surveys per year is to get at this detection probability. I think you need multiple surveys to help us estimate that. I think what happened in that case, in the bottom right, is that the species, the bird, was detected on quite a few of the other years, and so probably the model was like, well, it seems unlikely that it was present and then left and then came back and then left again and then came back. This seems less likely than it was there the whole time, but just the person missed it 2 years in a row or 2 years apart. Yeah. And then for elevation, these are the transition probabilities. So colonization probability on the left and local extinction probability on the right. And so you can see it seems like there's kind of a little bit of a nice Goldilocks thing with elevation. They seem to colonize or tend to favor these elevations between maybe 1,000 and 2,000 meters. And local extinction probability is just kind of decreasing overall. And again, here, we could have probably captured the left-hand side with quadratic effect and the right-hand side with something like a linear effect or something, but it's nice not to have to actually make that assumption about the shape of the relationship. And by the way, another comment about this, I think if we had done that, if we'd included, if we'd modeled the left with a quadratic effect and the right with a linear effect instead of having splines do the job, my guess is that we would get almost the same shapes overall, but thinner, narrower confidence bands. And I think that's because we're giving the model, we're making a stronger assumption about the shape of the relationship. So we're not giving the model as much kind of flexibility in estimating the shape of the relationship. And so the model could tend to be overconfident because you give it more information with this stronger assumption, and so it tends to be overconfident. All right, so my third example is on humans, but it's accelerometry data. So you could imagine that they're rabbits or sharks or whatever you work with. And I'm going to study their activity patterns through the day. That was like an actual published study where they were doing that. They put accelerometers, Fitbits, or something on people, and then looked at their activity through the day. And I'm going to use this analysis or application to illustrate a little bit what we can do with Bayesian inference in HMMTMB. So these are the kind of data that we have here on the left-hand side. This is, I don't exactly know how they got it, but the authors of the study got it, but it's basically a one-dimensional summary of acceleration, so kind of an overall activity level. And we have that at a 5-minute time resolution for 2 patients over a few days. And obviously, as you could imagine, there's like cyclical patterns through the day because people go to sleep and so on. So I'm going to model this with a 2-state hidden Markov model, where in each state, this activity is modeled with a gamma distribution. I'm going to try to use these 2 states to identify kind of low activity and high activity or something. I actually also need zero inflation here because the gamma distribution can deal with 0 values, and there are some values of activity that are exactly 0. And then to try to address the author's original question, I'm going to include the time of day as a covariate with a nonlinear cyclical effect. I don't know if anybody's Bayesian in the room, but the basic idea with Bayesian inference is that instead of just wanting to get point estimates and confidence intervals and so on, you want to get kind of posterior, you want to estimate the posterior distribution of parameters, and that's kind of a nice measure of uncertainty. And there is a way to do this in HMMTMB, and again, it's very much kind of based on other people's work. There's a package called StanTMB that translates TMB model objects into Stan model objects. And Stan is like a great package for Bayesian inference that uses MCMC to get posterior samples. So essentially, it gives you samples that approximate the posterior distribution for all your parameters. And there's a separate function to do that in HMMTMB. So once you've created your model object, you can choose the default route, which is like just uses TMB and kind of like this marginal likelihood approach I mentioned, or you can use the other route, which is, I think it's called fitStan, the other function that uses Stan to fit the model. There's some subtle nuances in exactly how the models are fitted, but I think in some cases there's good reasons to use the Bayesian approach. If you're already Bayesian, I'm sure you don't need, you're already convinced, but if you're not, there's a reason people do that. So what you need to specify a few more things if you want to go that way. You need to specify the number of NCMC chains, the length of the chains, and so on. So if you've used SEN before, you'll be familiar with that. And then you need to specify priors, and I'm not going to explain how I chose those priors, but you can specify in HMMTMB normal priors on the length scale of the parameters, which gives a decent amount of flexibility. And then the output that you get from this function is the posterior samples. So those samples, it's basically a whole bunch of numbers for each parameter that approximate their posterior distribution. And so for each parameter, this is the mean in the two states and the standard deviation in the two states for our activity distribution. You get not just a point estimate, but again this nice distribution of uncertainty. Those posterior samples you can also translate into posterior samples for more complicated things, like for example for the actual curves of the distribution. So here it might just look like thick lines, but for each color there's actually something like 50 lines that represent 50 posterior samples for the distribution of activity, the estimated distribution of activity in each state. And so it captures the uncertainty. It kind of gives you a visual way to assess the uncertainty on that estimated distribution, which seems pretty small here. As a side note, you might notice that there's a bump here on the right that we're not really capturing with this model. So this is typically something that you might be concerned about if you really want the model to capture the distribution of activity really well. But nonetheless, we capture kind of lower activity in the blue state and kind of higher activity in the red state. And then one other really interesting thing we can do with these posterior samples is also to get posterior samples of that relationship between the covariates and the transition probabilities, or here, these stationary state probabilities. So this shows us the probability of being in the active in red or inactive state in blue through the day, but with a measure of uncertainty. So these aren't like individual specific, like in the petrol example, these are posterior samples. So I could just have one line in the middle that shows us the kind of the point estimate of that relationship, but that would ignore all the uncertainty. And there's actually a decent amount of uncertainty in this case about exactly like at what time of day people start being more active and so on. Yeah, I had a little statistical detail to mention here, but I don't know that I have time and that it's interesting enough, so maybe I'll just conclude. So I mentioned quite a few things, but there's a lot more that's possible. So some of the other things that you can do in HMMTMB is you can include covariate effects on the observation parameters, so like on the mean activity within each state, or on the mean step length of the petrols within each behavioral state, or things like that. And that opens the way for a really general class of models that we call Markov switching regression. You can do things like more general dependent structures. So sometimes people complain that the hidden Markov model is a little bit too simple because it assumes that, for example, the animal's behavior at time t only depends on the animal's behavior at time t-1. But you might think, well, actually, I know that elk or whatever when they're foraging, they tend to forage for 4 hours at a time. So it should kind of have a longer term memory in some ways. And there's ways to do that in there's ways to essentially rewrite those more complex models into hidden Markov models. You do run into computational issues because you end up needing very big kind of state spaces and stuff to model these, but you can in principle do that kind of stuff. And then there's things like supervised learning that you can also do in the package in these models, where essentially you're giving some information to the model about the states that you're interested in. So that's in the case where you have direct observations of the hidden process for some subset of the data. You might actually be able to go on a boat and go and watch the petrols for like some short period, and you use that to somehow tell the model exactly what you're expecting the behaviors to look like and so on. You can also train a model on one data set and then use it on another data set to get like the state classification, and like all sorts of things like that are possible. There's quite a few like online resources. So I wrote maybe 6 or 7 vignettes with like examples in ecology, examples in other fields, and they're all available on GitHub, on the GitHub repository for HMMTMB. The package is also on CRAN, so if you want to install it, you can just download it or install it like any other package. And then if you're more interested in like the more kind of technical aspect and how it's implemented and so on, some of that like marginal likelihood stuff, This is all described in the HMMTMB paper that's on arXiv and that was recently accepted in the Journal of Statistical Software. One small comment I should make for those of you who were at my little workshop yesterday is that this package, this is a different package to the package I mentioned yesterday. Yesterday I talked about momentum, and this is HMMTMB. And to some extent, they use different syntaxes and stuff, which might be a little bit confusing. But the workflow, the modeling decisions, the output, the interpretation of the results, like everything is the same. Like there's small, well, I guess big syntactic differences, but from a statistical perspective, they're very much the same models and with the same interpretation. So if you learned something yesterday, it's not completely useless if you decide to use HMMTMB at the end of the day. And that's it for me. Thank you.
Thank you so much. Any questions in the room?
Hey, thanks, Theo. I thought that was really great. So many ideas of how to get at biological inferences that I want to get at, but are hidden from me. I had a question about the petrels, and I don't know, maybe the solution is in the Bayesian approach. One of the things that I like to do, for better or for worse, is to have these hidden states, come up with a way to assign them to some behavior, and then look at the individual differences. It gets us into that world of best, least, unbiased predictors, BLUPs, that people are pretty critical of. The main criticism of those BLUPs in the beginning is this, that if you use frequentist approaches, those BLUPs are forced into a normal distribution, which doesn't necessarily represent the distribution of the trait. So it works in a way to, as you say, mop up variance you don't want to deal with. But if you want to then do further stats on those individual differences, one needs to account for the certainty in your individual estimate, and also let those estimates at the population level have a shape that are something other than normal. Can you imagine, I guess, is the Bayesian approach applicable to the petrels? Is that a solution, or would you imagine more generalized error distributions for those individual differences?
Yeah, okay, thanks. Great question. I mean, so in the petrol example, the random effect, is like the intercept. So essentially the kind of how much each individual is allowed to deviate from that population mean is normally distributed, and that is a pretty strong assumption. And in HMMTMB, that you kind of have to have that assumption. Whether you're Bayesian or not, when you're Bayesian, random effects are treated a little bit differently, but they still have the same model assumption of normality. Yeah, so one thing you could do, well, you could have fixed effects. So that's the case where you estimate the individual level differences, completely separately from each other. That becomes very, intractable if you have like, tens or hundreds or like a really large number because that's so many parameters to estimate. One thing that I really like to do, but is not always an option, is to include individual level covariates that, instead of including, if I had more information about the individuals, like the sex and age and size and whatever else informs their behavior, it would be better to include these as effects on the transition probabilities, because it would kind of somehow mop up that individual variability, but actually with like kind of a mechanistic flavor to it, where it's actually telling me, well, this individual spends more time foraging, but it's because it's younger and so it has more time for foraging or something. Which this is ignoring. This is very much the case where you have no information, you just want to do you want to somehow account for it, but without any structure. So yeah, I think this probably has some of those flaws of those kind of normally distributed random effects that you're mentioning, and I think it would be difficult to get away from that. Yeah.
Hey, Theo, thanks for a great talk and kudos on the very powerful package. I just have a question about another maybe statistical difficulty that you probably thought about. And so I'm curious to hear your thoughts on it. When it comes to observations, we often have observations that are imperfect. I'm thinking about telemetry studies where we might have positions with huge positioning errors, or geolocation studies, for instance. And there are state space models that try to filter out those observation noise, and the latent state is the actual location, it's unobserved, just inferred from our problematic observations. I'm wondering if HMMTMB has built into it a way to account for those observation errors, or is that still a statistically very difficult, nut to crack, because you're dealing with two latent unobserved states layered on top of each other? And so just curious about your thoughts on that. Thanks.
Yeah, no, I mean, great question. And yeah, so HMMTMB assumes no measurement error and assumes regular time interval. So this is stuff you would have to do prior to that. And there's other R packages that I'm sure you're familiar with, that fit those state space models to deal with it. My, career dream would be to have those like state space hidden Markov models, kind of everything integrated, but that's super complicated to actually do in practice. So what people do is, as you've mentioned, it's like fancy interpolation, right? They try to regularize the track, they try to kind of somehow filter out the measurement error. One problem with this is that then you end up with locations that you have to assume are the true locations of the animal, even though they're not. They're the output of another model, of a state space model. And so you're kind of failing to propagate that error. And so one kind of intermediate approach that has been used, that is not perfect either, but is better than that, is called multiple imputation, where you generate not just one plausible track, but like 50 plausible tracks that could have given rise to those observations. And that is a way, at least if your kind of imputation model is good, that is a way to capture the uncertainty that there was in these like regular predictions. It's very computational because it means you have to fit 50 hidden Markov models, and then there's formulas to combine the results from these 50 hidden Markov models in such a way that you get standard errors that account not just for the uncertainty in each model, but the uncertainty kind of across models. So yeah, maybe that's one direction to explore. And there's packages to do that, and not this one. There's other packages to do that. Yeah.
Yeah, I think you commented on this, but I wanted more explanation to that. So when your model cannot capture the distribution of your observation, how do you make sense of the model? Like, do you still go ahead and use the model and make prediction, like study the model? How do you make sense in general about the model that is not able to capture?
Yeah, no, great question. So are you referring maybe to something like this? Like, yeah, something like this. So here, yeah, we have two distributions. And if you think about, like putting these distributions on top of each other, we're not quite capturing, for example, this bump here. There are ways that you could make this better. You could add one more. The most obvious thing to do here would be to add one more state, and then you would have a third, basically that adds one more distribution to capture kind of the shape of that overall distribution. So kind of one more mixture component for more flexibility. But maybe more to the point of your question, this lack of fit here isn't necessarily concerning. And the reason why it's not necessarily concerning is that if what you're really interested in is the dynamics of the underlying process, and that's often what you're interested in, what you want to make sure is that high activity states or high activity observations are grouped together correctly, and low activity observations are grouped together correctly. But my guess is that this model, despite lack of fit, is still going to do that. It's still going to, even if it doesn't exactly capture the shape of the data, it still has a pretty good sense of it's going to have a cutoff somewhere here for these are low activity observations, these are high activity observations. And so if you trust that, more like that inference about the state process, then you might be pretty happy with this inference, because the correctness of the relationship here doesn't rely on matching the distribution of the observation perfectly, but just on having a pretty good understanding of the dynamics of that hidden process.
Just going to check the chat. Any more questions in the room before we wrap up? Thank you so much.
Thank you so much.
There's still lots of snacks, I believe, so help yourself on the way out.
Thanks so much.